أهلا بيكم في قناة الأحياء للصف الثالث ثانوي. In today's lecture we're going to talk about flowers. So let's begin. Reproduction in flowering plants. The flowering plants, large group of seed plants. Their seeds develop with a pericarp. Pericarp معناها القشرة. هو ده اللي هيبقى قشرة الفاكهة بعد كده. It's called angiospermy. The flower in these plants is the specialized organ for reproduction. Now the flower arises from the region called the axil. axil اللي هو الإبط، يعني الإبط اللي بين الورقة، بين ورقة النبات والجذع اللي هي ماسكة فيه. الحتة دي بيسموها bract. Okay, so the flower arises from the axils of either a green or a scale leaf called bract. In some cases, the flowers come out without bracts. Flowers may be solitary apical, يعني على القمة كده زي التيوليبس، وبسبب انها موجودة على القمة يعني هتمنع ان النبات ده ينمو بعد كده. It stops growth of the stem. او ممكن الزهرة تطلع من الجنب، من جنب محور النبات، called solitary axial, as in petunia. Okay, in other cases flowers may be grouped. ممكن يبقوا عبارة عن مجموعة of flowers grouped on the floral axis into various aggregations called inflorescence، زي البينز كده، زي الفول والمنثر. Okay, parts of the flower. Now I want you to pay attention to this, please. We said that it emerges from the bract at the axil of a leaf. A typical flower that we will study has 4 floral whorls or layers. Okay, whorl يعني layer دائرية كده. The first one is called the calyx. It's the outer whorl of the flower, formed of green leaves known as sepals, as you can see here. And the function of this calyx is to protect the inner parts of the flower against drought, rain, or wind. The other layer is corolla, and maybe a layer or 2. It's composed of one row or more of petals. And the function of the corolla, it helps in protection of the flower's sexual parts, like the ovary, for example. Okay, and also attract insects for the pollination process to occur. So the function of the corolla, the first layer is the calyx. It's for protection. And the other layer is the corolla. It's for protection also of the flower's sexual organs, sexual parts, and they attract insects as well. This is, demonstration of the structure of the flower. You see, this is the ovary here. It has a style, and then the tip of the style is called the stigma. Stigma, style, ovary. This is the female part, of course. And the male part, it has a filament, as you can see here, filament with an anther on top. An anther of top is it's 4 sacs of germ tissue there to be liberated later on. So, by the way, this is a bisexual flower. It has both the ovary, the female, and the male. Not this doesn't occur in all flowers. Some flowers, they have only the male parts. Some flowers only have the female parts. Some flowers have a well-developed female rather than the male parts, so matured more than the male parts. Okay, and this will be of significance later on, as we will explain. But the typical flower that we will study has both parts, the male parts and the female parts. And remember, the male parts has the anther, okay, and has the filament. The female part has the ovary and the style, and the tip of the style is the stigma, and pollination will occur at this region in the stigma. Okay, androecium is the male organ, consists of stamens. Each stamen consists of a filament which carries an apical anther and that contains 4 sacs of pollen grains. I told you it has 4 sacs of pollen grains in it. The gynoecium, which is the female organ, is the central whorl of the flower and consists of 1 or more carpels. It's called altogether the carpel. The carpel base is swollen and called the ovary, which contains the ovules inside it. The carpel may fuse or remain separate and enclose 1 or more lobules. Okay, there's a thin neck after that. It's called the style, and then the tip of the style ends by a sticky disc called stigma. Why sticky? For the pollens to be attached to it. Okay, of course they travel through air, they get on the stigma, they get stuck to the stigma by this sticky substance. Okay, what are the functions of the flower? First, formation of pollen grains. A transverse section in a mature anther, which is this part, which is the tip of the male part. There are 4 sacs of pollen grains. During flower development and before formation of pollen grains, these sacs are full of large nucleated diploid cells called the spore mother cells. Again, so it has 4 sacs full of pollen grains, and these sacs full of large nucleated diploid cells, 2 end cells called the spore mother cells. Now each of these cells is divided meiotically, not mitotically, meiotically, to form 4 haploid number cells called microspores. So the spore mother cell, which is a 2N, divides by means of meiosis to form 4 haploid parts called the microspores. Each of these microspores develop into a pollen grain through a mitotic division to achieve the number of its nucleus, and the mitotic division occurs in the nucleus, and the nucleus divides into 2 nuclei called, one of them is called the tube nucleus and the other is called the generative nucleus, and each of them has a purpose. Remember, the microspores develop into a pollen grain through mitotic division, and the nucleus, by this means of mitotic division, divides into 2 nuclei called the tube and the generative nuclei. The tube and generative nuclei. See that? 2 nuclei. The wall of the pollen grain then thickens for protection. Of course, now you know whenever the wall is thickened, this means protection. And the anther matures, and the wall in between the adjacent pollens, becomes degenerated, and the sac open. This will lead to release of the pollen grains that become ready for dispersal. Now, in the exam, you're asked to draw the stages of formation of pollen grains. Now, please, pay attention to this, because in the exam, you may confuse formation of pollen grains with pollination of the grain. Okay? So you should read the question carefully and know what is requested precisely. Formation of the pollen grain or pollination of the pollen grain. The formation of the pollen grain means the formation, the process that the pollen grain forms, not pollinates the ovary of the plant. Okay. After they mature, the sacs open and the pollen grains are released and become ready for dispersal. Now, formation of the ovules, on the other hand, it starts by a simple swelling on the interior ovary wall. Contains a large number of large spore mother cell. Large, yes, in size, spore mother cell, and during the growth of this ovule, a funicle develop. Funicle, means a little passage. Funicle develop connecting it with the ovary wall, زي الحبل السري كده اللي بيربط الجنين بالرحم. Okay. بالمشيمة اللي في الرحم. فهنا الفونيكل ده علشان يربط الاوفيول بالاوفري وول. اوكي. فايه اللي بيحصل؟ ايه الفانكشن of this follicle through which the food material reaches it. Okay. Two integuments surround the ovule completely, and there is a minute hole called the micropyle, as you can see here. The micropyle through which the ovule is fertilized. So through the funicle, it will get its food. Through the micropyle, it will be fertilized. Okay? Again, through the funicle, they will get its food, and through the micropyle, it will get fertilized. Okay, within the ovule, a mother spore cell, which is diploid in number, divides meiotically and gives a row of 4 haploid cells, 4 in number. 3 of them degenerate, and the fourth one grows rapidly, forming the embryo sac. So 3 degenerate, and 1 of them forms the embryo sac, surrounded by nutritive called the nucellus. Nucellus will feed the embryo sac, then the embryo. Then the following happens. The nucleus divides mitotically 3 times. When you count this 3 times, it will produce 8 nuclei. As you can see here, 8 nuclei. Okay? 2 of these nuclei will migrate to the center, called polar nuclei, and each of the remaining 3 nuclei on each side of the sac becomes enveloped by some of the cytoplasm and thin membrane, forming distinct cells. So the nuclei will be 8 in number. 2 of them will migrate to the center, 3 on one side and 3 on the other side, and each of these 3 acquires little cytoplasm and a membrane to form an individual cell or distinct cells. Now, the middle of the 3 cells that are close to the micropyle, the middle one, will grow into the egg. Remember this, the middle one will grow into the egg, while the 2 sides, called synergids or helper cells, how can they help? They help in directing, the pollen to fertilize the egg. Okay? So the 3 cells that are formed near the micropyle. The central one will become the egg, and the two surrounding cells will become synergids or helper cells that will help in guiding the pollination process. Now, the three cells on the other part of the micropyle, or distant from the micropyle, they are called the antipodal cells because they are on the other side. And once all these cells are formed, the ovule is ready for fertilization. Okay? So again, the nucleus divides mitotically 3 times, gives 8 nuclei. 4 of them migrate to each pole of the embryo sac. Now, from the 4, 1 moves on each side, 1 moves to the center, so 2 at the center. They are called the 2 polar nuclei. And the remaining 3, the remaining 3 on each side acquires cytoplasm and the thin membrane to form individual cells. And the 3 near the micropyle, the central one will become the egg. The surrounding ones are helper cells or synergid cells that will guide in direction of pollination. And once all these formed, the ovule is ready for fertilization. Now, what happens next? Of course, the next process is pollination and fertilization. Pollination is the process by which the pollen grains transported from the anther, which is the male part, to the stigma, which is the female part of the flower. Types of pollination. There are self-pollination and cross-pollination. Self-pollination, from the name, it occurs in the same flower. Okay? Or on another flower, but on the same plant. So either the pollination that occurs in the same flower or to another flower on the same plant. It's not another plant. All this is called self-pollination. Cross-pollination, pollen grains transported from the flower anther of a plant to the stigma of another plant. Now I want to ask you a question here. Which is more effectful, the self-pollination or cross-pollination? I think it's the cross-pollination because this ensures diversity, because this happens between 2 plants, so different genetic materials, so means diversity. Diversity occurs more in cross-pollination. Okay? But of course, the other plant must be of the same species. Now, pollination may occur in plants according to the presence of certain factors. For example, if a flower is unisexual, so the pollens has to go to another flower on the same plant or another flower on another plant. Okay, but in this case we're talking cross-pollination, so it's on another plant. Or maybe the organs of one sex mature before those of the other sex, so unable to perform self-pollination as well. Or maybe because of the height of the anthers is lower than the stigma, so it will make it so difficult to be pollinated from on the same flower. Cross-pollination means to be transported from one flower to another, and these means of transportation are air, insects, water, or even in man. For air, of course, they must be very light in weight. For insects, they must be rather, rough to be able to stick to the insect's body, or through water, or even through man, okay? Artificially through man. Now, fertilization. I told you before to pay attention. There is a difference between formation of the pollen and pollination or the process of fertilization. Now, the pollen grain germination, when the pollen grains fall on the stigma by any means of transport, they germinate and the tube nucleus forms the pollen tube. Remember the tube nucleus we talked about earlier? The tube and regenerate nucleus, or generative nucleus. The tube nucleus forms the pollen tube, which penetrates the stigma and crosses through the style till it reaches the ovule's micropiles. So see all this distance, the tube formed by the nucleus. Okay, this tube penetrates the stigma, penetrates the style, and goes all the way to the micropyle to fertilize the egg. Then the tube nucleus after that degenerates because the tube is already formed, while the generative nucleus is divided one mitotic division. Again, pay attention please. After the tube nucleus has done its work, it degenerates. What about the generative nucleus? The generative nucleus divides by one mitotic division into two male nuclei. One male nucleus, which is a haploid in number, fuse with the egg, forming the zygote. Okay? And the zygote becomes 2n, of course, because the haploid numbers, they fuse together, and then start to divide, forming the embryo, 2n. Again, the generative nucleus divides by one mitotic division into two male nuclei. One male nucleus fuses with the egg, so fertilizes the egg. A haploid and a haploid means diploid number, so the zygote is diploid number. Okay? And then it starts to divide to form the embryo. So male nucleus, haploid, plus the egg nucleus, haploid, forms the zygote, which is a diploid, that forms the embryo, which is a diploid. What about the second male nucleus? The second male nucleus fuses with the two central nuclei, the one we call the nuclei of the embryo sac in the middle. See? Forming the endosperm nucleus. Of course, one haploid plus two plus diploid, haploid plus diploid means triploid. Formation of a triploid part. This is why it's called triple fusion. So male nucleus plus two nuclei of the embryo sac will produce, this male nucleus is haploid, the two nuclei of the embryo sac are diploid. The endosperm nucleus result will be triploid, 3n. Remember this. Okay? Because of the one mitotic division that happened and two male nuclei originated, this is called double fertilization, because we have two male nuclei, one fertilized the ova and the other went to the forms the endosperm tissue. The endosperm nucleus then divides and forming the endosperm tissue. This supplies the early developing embryo with food, and this tissue may remain outside the embryo, occupying a part of the seed. Okay, I hope you understood the process of fertilization. You know, it goes all the way to the micropyle, fertilizes the egg. One mitotic division produces two male nuclei. One fertilizes the egg and the other forms the endosperm tissue by the, going to the central nuclei. And remember that the endosperm tissue or the endosperm nucleus is 3n. Okay. Formation of fruit and seed. The embryo may keep the endosperm. In this case, the seeds are called endospermic seeds. in which they have only one cotyledon, and the integuments, cotyledon means فلقة, ذات الفلقة الواحدة, in which the integuments of the ovary and ovule fuse together, forming a single-seeded fruit. Now this occurs like you see in maize and wheat, زي في القمح مثلا. The embryo may feed on the endospermic tissue, okay, during its development, and the plant has to store another food in the cotyledons. That's why another or two cotyledons occur. This will be called dicotyledons, where the integuments of the ovule harden, forming the seed shell, or called testa. It's called seed and not grain. Then it's called seed, as in beans and pea seeds. After fertilization, the calyx, the corolla, the androecium, and the style and the stigma wilt and fall out. Only the ovary remains that stores food. It ripens and transformed into a fruit due to the hormones auxins secreted by the ovary. So the rest of the parts will be wilt and fall out. The ovary's wall is transformed into the pericarp, بيتحول للقشرة بتاعت الفاكهة, and the seed wall into the seed coat or testa. يبقى okay, the pericarp هيتحول للقشرة اللي هي بتاعت الفاكهة, is seed wall into the seed coat or testa. The two synergid cells and the antipodal cells, the ones we talked about during the formation of the what happens in the ovary, disappear. These cells disappear, while the micropyle remains, so as to allow water to get into the seed during germination. Now, the micropyle, we said its function to allow for fertilization, okay, during pollination. The other mission or the other function is to allow water after fertilization occurred, and to allow water to get into the seed during germination. There are some fruits which keep some parts of the flower. For example, leaflets and calyx are spared in pomegranates, زي الرمان مثلا. The calyx may take part as an eggplant, زي البتنجان. The calyx بيبقى موجود زي انتوا كلكم بتشوفوا اللي هي الحته الخضراء عند قمه البتنجان دي. وفي البلح برضو in dates. The corolla leaflets may stay as in marrow fruits زي في الكوسه مثلا. The corolla leaflets موجوده هناك, بتفضل موجوده, يعني ما بيحصلهاش انها wilts and disappear and fall out. Okay, there are fruits called false fruit, in which the fruit, any part except its ovary, enlarges to store food. Okay, so other parts rather than the ovary enlarge to store food, like what we see in apple. Okay, there is a phenomenon called parthenocarpy. Parthenocarpy is the natural development of fruit devoid of seeds. Devoid of seeds means no fertilization happened. Okay, such an example is a banana. Banana and pineapples are such fruits. They can be carried out artificially, also by spraying the stigma with an extract of pollen grains. بيطحنوا pollen grains وبيحطوها في ether solution or indole or naphthol acetic acid to stimulate the ovary to form the fruit. Now, the maturation of the fruit and seeds often leads to discontinuity of the plant growth and sometimes to its death. Why? Due to consumption of stored food and inhibition of hormones. Okay, if pollination and fertilization did not take place, the flower withers بتدبل يعني and drops off without fruit formation. Well, I hope you got the maximum benefit from this lecture, and thank you very much for watching.